Dear friends, today we are going to introduce the elementary idea regarding the microfossils. Microfossil studies allow precise local, regional and global time stratigraphic correlations that help in the hydrocarbon exploration and trend delineation, that is regional stratigraphic and geological studies and exploration evaluations. Analysis of the microfossils helps us to recognize the paleoenvironmental distributions, which in turn is useful to interpret sequence stratigraphy and reconstruct the paleogeography and paleoclimate. Some microfossils function as paleothermometers by undergoing irreversible color changes with post-burial heating. As such, they indicate hydrocarbon maturity levels. Microfossils useful in hydrocarbon exploration can be divided into 5 principal microfossil groups, that is based on the composition of the shell, test or hard parts. Composition, fossil groups which are calcareous, they include foraminifera, ostracods, calcareous nanofossils. Now agglutinated, it includes foraminifera. Siliceous includes radiolarians, diatoms, silicoflagellates, while phosphatic microfossils are conodonts. Organic wall microfossils includes chitinozoans, pollens, spores, equitarchs, dinoflagellates, which are collectively known as paleonomorphs. Calcareous microfossils have shells composed of calcite or aragonite. These organisms are present in most marine and in some non-marine environments at great oceanic depths, characterized by low temperature and high hydrostatic pressure. However, calcareous remains are largely or completely dissolved. The depths below which this occurs, which varies in different oceanographic settings, is termed the carbonate compensation depth, and CCD for short. There are 3 principal types of calcareous microfossils. Number 1, calcareous foraminifera, number 2, ostracods, and number 3, calcareous nanofossils. Coming to calcareous foraminifera, calcareous foraminifera are a group of unicellular organisms, that is protists, that secrete a rigid calcite or aragonite shells or tests. Fossils of these forms are found in sediments of brackish to marine origin, and they originated from Silurian, and it ranges up to the Holocene in age. Most are benthic, that is bottom dwelling, but a significant group in the late Mesozoic and Cenozoic are planktonic, that is floating forms. Some stratigraphically important foraminifera developed complex internal structures and frequently large test size. Studied primarily in thin section, these include the fissulinids that ranges from Pennsylvanian to Permian, and several groups of so-called larger foraminifera that have a range from Triassic to Holocene. They occur primarily in carbonate or fine-grained clastic rocks and are excellent time markers. Since many species have limited and well-known environmental ranges, therefore they are excellent paleobathymetric and paleoenvironmental indicators, especially in the younger Phanerozoic rocks. Ostracods are microscopic crustaceans whose fossils are found in Cambrian to Holocene. They occur in most marine and non-marine depositional environments and are generally excellent environmental indicators. The paleontologic application of the ostracods is limited because they are in many sections very rare, and number 2, many species are endemic to local basins. So, their age and environmental ranges are poorly understood. Ostracods typically have rapid evolutionary rates and are useful in biostratigraphic tools in some situations, that is, in Paleozoic sequences, in marine environments where wide ranging species are present, and for local stratigraphy in basins of limited extent, and lacustrine environments where they are frequently one of the few microfossils present. Ostracods may also indicate thermal maturation of source rocks. Now, coming to calcareous nanofossils, the term calcareous nanofossils include both fossil coccoliths and nanoliths. Coccoliths are minute, that is less than 25 nanometers, calcite forms produced by unicellular marine plants, that is golden brown algae. The origin of nanoliths is uncertain, but these calcite bodies are associated with fossil coccolith assemblages in marine sediments and are also organically derived. Calcareous nanofossils are an excellent biostratigraphic tool because of their rapid evolution and geographic dispersal, that is, their entire life cycle is in the photic zone of the ocean, as well as their varied and distinct morphologies. The oldest known calcareous nanofossils are late Triassic. They are a crucial microfossil group in calibrating the Jurassic Holocene marine record. Relatively little has been published about the paleogeographic distributions of calcareous nanofossils. Less is known about their exact paleoenvironmental preferences, although they have been shown occasionally to penetrate into the shallow marine environments. Their main industrial application is their calibration to published time scales and sequence stratigraphic records, especially the association of high abundance with condensed marine sections. Coming to the agglutinated microfossils, agglutinated or arenaceous microfossils have tests or shells constructed from sedimentary particles bound together by organic, calcareous, siliceous or ferruginous cement. There is only one type of stratigraphically significant agglutinated microfossil. Agglutinated foraminifera are benthic microfossils found in rocks of Cambrian through Holocene age and in most marine and brackish environments, particularly in clastic facies. They construct their tests by gluing sedimentary grains together in contrast to the other types of foraminifera, which secrete their test. The sedimentary particles used by these forms may include silt or sand grains, glauconite, sponge spicules, or even other foraminiferal tests. Some species are highly selective in the material used in its arrangement, although they are sometimes useful in age and age determination. Agglutinated foraminifera are especially valuable as paleoenvironmental indicators because they are particularly characteristics of very shallow marine to brackish environments and very deep environments. Agglutinated foraminifera are the dominant and distinctive microfauna in the Cretaceous and Tertiary flesh species. The color of agglutinated foraminifera has recently been shown to alter irreversibly with heating. both naturally within buried sediment and experimentally also. Siliceous microfossils are protists with shells constructed of opaline, that is amorphous silica. There is no intense dissolution of siliceous remains in the deep ocean. Sediments deposited below carbonate compensation depth are commonly enriched in silica by removal of the carbonates, sometimes to the point of forming siliceous oozes. With subsequent remobilization of the silica, deep sea cherts may be formed. Siliceous microfossils are subject to burial diagenesis and become rare at great well depths, except when recrystallized, preserved in nodules or concretions, or replaced by pyrite or calcite. There are three major groups of siliceous microfossils, that is radiolarians, diatoms and silicoflagellates. Radiolarians are planktonic protists that occur primarily in open marine deep water settings. They are useful time indicators and are found in rocks of Cambrian to Holocene in age. They may be the only common microfossils in abyssal environments, commonly forming radiolarian oozes, radiolarian chert. The product of silica diagenesis is fairly widespread in the geological record. Radiolarians are common in some marine source rocks. Now coming to diatoms. Diatoms are photosynthesizing protists that occur in both marine and non-marine environments. Marine diatoms range from late Jurassic or early Cretaceous to Holocene and are particularly useful for age and environmental determinations in the late Cenozoic. Non-marine diatoms range from Eocene to Holocene and also are useful in the late Cenozoic. These microfossils can be a major rock forming group, forming sedimentary rocks, that is diatomite, consisting primarily of diatoms. Diatomaceous sediments, when altered by burial diagenesis, are converted into siliceous shale, porcelainite and chert. Such rocks can serve as sources and fractured reservoirs for hydrocarbons, for example, Monterey Formation of California. The changes in rock properties associated with silica diagenesis permit seismic definition of silica phase transformation zones in the subsurface, for example, bottom simulating reflector. Silicoflagellates. Silicoflagellates are another group of planktonic photosynthesizing marine protists. They commonly occur with diatoms. Silicoflagellates range in age from Cretaceous to Holocene. Although not as common as diatoms, they are useful time indicators, particularly in the late Cenozoic. As a group, they were much more abundant during the early and middle Cenozoic than today. They have been used to estimate marine paleotemperature in the late Tertiary and Quaternary. The next group of microfossils is phosphatic microfossils. Phosphatic microfossils, notably conodonts, are composed of crystallites of calcium carbonate and calcium phosphate, that is apatite, embedded in an organic matrix. There is one type of stratigraphically significant phosphatic microfossils, that is conodonts, but fish teeth of less practical utility are found in some marine strata. Conodonts are extinct tooth-like microfossils composed of calcium phosphate whose biological affinities, while poorly understood, lie with chordates. Conodonts are widely distributed in marine rocks of Cambrian through Triassic age. They are excellent indicators of time and thermal maturity, especially in carbonates. Whether or where other methods of evaluating organic thermal maturity are less successful, conodonts are commonly used as zonal indices for the latest Cambrian through Triassic because they were abundant, evolved rapidly, and were widespread geographically. Although found in most marine rocks, conodonts are most efficiently recovered from the insoluble residues of carbonates dissolved in weak acids or from easily disaggregated shales. Individual conodonts vary greatly in morphology, and taxonomy was originally based on the morphology of these individual specimens. While conodonts are common, the preserved remains of the soft-bodied animal that bore them are extremely rare. Conodonts appear to have been located in the cephalic area and may have functioned as teeth. However, the conodont animal apparently bore many conodonts of differing shapes and morphologies. Based on the study of very rare whole animal specimens and a rare bedding plane grouping of conodonts representing individual animals. This recent information has led to more accurate multi-element species concepts. The next group of microfossils is organic walled microfossils. Organic walled microfossils are composed of entirely unmineralized proteinaceous material. There are 4 types of organic walled microfossils, starting from chitinozoans, spores and pollen, acritarchs, and dinoflagellates. Starting with chitinozoans. Chitinozoans are marine organic walled flask-shaped microfossils that ranges from 50 nanometer to 2 millimeters or 50 micrometers to 2 millimeters in size, that occur in rocks of Ordovician to Devonian age. The biological affinities of chitinozoans are poorly understood, but they may be eggs of marine metazoans. They are excellent biostratigraphic indices and useful paleoenvironmental markers. They also have potential as thermal maturity indices. The next group is spores and pollens. Spores and pollen are parts of the reproductive cycles of plants and range in age from late Ordovician and Carboniferous respectively to Holocene. Although land derived, the grains can be carried by wind and water currents into a marine and non-marine, that is particularly lacustrine and fluviatile environments. The type and relative abundance of spores and pollen provide useful paleoenvironmental and paleoclimatic information, and they are widely used for basinal and regional stratigraphic correlation. Spores and pollen are also very useful in estimating thermal maturity, especially at temperature levels associated with hydrocarbon generation. The next group is acritarchs. Acritarchs are marine microplankton of unknown biological affinity ranging from Precambrian to Holocene in age. They are excellent biostratigraphic indices for Proterozoic through Devonian strata, but are less important in Mesozoic and Cenozoic. Acritarchs occur abundantly in fine-grained rocks and are geographically widespread. They have been used for paleoecology, paleogeography, and thermal maturity. The next group is dinoflagellates. The dinoflagellates are resting cysts of marine unicellular red algae. They occur abundantly in Upper Triassic to Holocene sediments and are excellent biostratigraphic indices because of their rapid evolution and widespread geographic distribution. Dinoflagellate cysts occur predominantly in marine rocks, but are also present in Cretaceous and Cenozoic, that is lacustrine facies. The morphology and diversity of dinoflagellate assemblages can be used to differentiate marine environments. Paleontologists have traditionally used fossil data to provide information about the relative age, stratigraphic correlation, and paleoenvironment, notably paleobathymetry of sedimentary strata. The importance of these tasks remain high, but new approaches and technology have extended the applicability of biostratigraphic data to analyzing thermal maturation and sequence stratigraphy. Coming to the biostratigraphic correlation and age determination. Changes in the abundance or species composition of fossil assemblages within a biostratigraphic zone are useful in refining correlation. Ratios or percentages of in situ versus revoked calcareous nanofossils or paleonomorph species may differentiate among distinct sediment packages. Generally, where in situ fossils are relatively abundant, revoked fossils are less common. High resolution methods are quantitative approaches that, in addition to species ranges, use subtle paleontological changes. For example, fossil abundance and diversity peaks. These data can be generated from actual counts of the species abundance or from estimates of relative abundance. This detailed, time-consuming approach provides closely spaced correlations that are particularly effective for sequence stratigraphy studies. Local or field correlation of reservoir units or any problem where detailed resolution is vital. The use of multiple fossil groups in some sections can greatly increase both the resolution and the level of confidence of the analysis. In the absence of index fossils, for example, within areas of high clastic dilution or when extending a chronostratigraphic horizon across environmental boundaries, correlation methods other than traditional use of fossil extinction plays an important role. Those frequently involve characteristics of the assemblage as a whole. Changes in the abundance or species composition of fossils assemblages within a biostratigraphic zone, and ratios or percentages of in situ versus revoked calcareous nanofossils or paleonomorph species. An example of the first type is the thin interval in the lower Pliocene, characterized by high abundances of the planktonic foraminiferal genus, which represents a set of paleogeographic conditions that can be correlated in many areas of the Mediterranean assemblage. The assemblage data can also help us to recognize diagnostic elements of stratigraphic sequences. Determining absolute ages through physical or chemical techniques such as radioisotope analysis, magnetostratigraphy, or fission track dating is not by itself a paleontological application. Approximate absolute ages can be derived for fossil assemblages and strata. A number of timescales have been published relating absolute age to the established sequence of primarily planktonic fossil events. These timescales differ somewhat in absolute ages assigned to the various fossil events, that is, inceptions and extinctions. In most applications, the consistent use of a single timescale is more important than the choice of the scale. Although absolute ages are not necessarily critical for well correlations, they are vital in studies that rely on determinations of the geological rates. The aim of the paleoenvironmental analysis or paleoecology is to reconstruct the biological, chemical and physical nature of the environment at the collection site at the time of the deposition based on the rock's paleontological record. Information can be reconstructed for depositional environments, paleobathymetry, position of ancient shorelines, paleoclimate, degree of oxygenation of the bottom water and sediment, and salinity of the waters. Paleoenvironmental knowledge is established by number 1, using the known tolerances of taxa or their nearest living relatives in modern environments, and number 2, studying the distribution of fossil taxa in well controlled ancient examples. The data are interpreted on the basis of this established paleoenvironmental knowledge.